Hello everyone. Today we will be presenting our capstone research on the effects of heavy metals on soil macroinvertebrates. More specifically, we wanted to look at how the heavy metals left behind by the discontinued Carey blast furnace at Rivers of Steel have impacted the soil macroinvertebrate communities.
Soil macroinvertebrates are very important in maintaining healthy and productive soil. Examining which species of macroinvertebrates are present within the soil can indicate whether the soil is healthy, contaminated, or overused. Biological diversity indices can also help estimate soil conditions to determine if further remediation options are necessary to restore the soil in these areas.
To determine the health of the soil, we completed both chemical and biological analyses of soil within the Rivers of Steel site as well as the designated control site within the parameters of our campus biotrails. 10 by 10 by 10 centimeter soil samples were collected and transported back to the lab in plastic bags. The soil was then placed into Burley's-Telgren funnels, a simple device used to collect macroinvertebrates using heat lamps and mesh screens. Once collected, macroinvertebrates were identified and tallied per order.
The chemical methods included pH, nitrogen, and phosphate soil test kits, as well as inductively coupled plasma optical emission spectroscopy, also known as ICP-OES, to test for lead, iron, zinc, and cadmium. Our results showed that heavy metal concentrations were much higher at the steel mill than the control site, and that pollution decreased as the distance from the blast furnace increased.
Biologically, Columbella and Acarina were the most prevalent macroinvertebrate communities identified. Communities generally decreased as pollution increased. A higher evenness at the Rivers of Steel site was reported. However, other biological indices were inconclusive. We noticed that the weather conditions played a significant part of our study, resulting in higher macroinvertebrates found following a precipitation event.
Chemically, the pH of the ROS site was much higher than the control, but this was due to the limestone, which is alkaline, being put on the soil. There were no statistical differences between the ROS and the control for phosphorus or nitrogen, so those can be rolled out as possible confounding variables. Further research must be done to draw significant conclusions based on our data. We would like to thank Dr. Diana Ortiz, Dr. Patrick Krantz, Dr. Helen Boylan, and Dr. Mark Wilson at Westminster College, as well as Ron Baroff from the Rivers of Steel for helping us along the way.